my phone rang, so it actually cut me off. So we're talking about the features of meiosis. When we talk about the feature of meiosis, there are two cell divisions. When we talk about homologous chromosomes, when you have one set from your dad and mom, these are going to replicate, and when you replicate them, you call them sister chromatids of homologous chromosomes. Well, these sister chromatids of homologous chromosomes become very closely attached, which is called synapsis. Now, the next thing that we are going to discuss is something that's called crossing over, which is a genetic recombination or a physical exchange that's going to occur between your non-sister chromatids. So I'm going to separate these out. When we talk about crossing over, a section from your mom's chromosomes is going to overlap a section of your dad's chromosomes, like this. Now, they are going to be closely attached, or a little closer from synapsis, but I need to separate them out a little bit to talk about this. Now, your mom and your dad's overlap. Now, that top section, when you have crossing over, what occurs in crossing over is there's a physical exchange of genetic material. And the piece that is actually being exchanged has a name called the chiasmata. So you have your dad's sister chromatids of homologous chromosomes, and a physical piece has exchanged. So a tiny little piece of your mother's genetic material attaches at the top. Now, the same thing happens to your mother's sister chromatids, where that there is a physical exchange between non-sister chromatids. That is crossing over. Now, crossing over is going to be what gives us genetic diversity. So, this is going to give genetic diversity. And then the other thing that I need to add on here is the site of crossover is called the chiasmata. All right. Other things that are different between mitosis and meiosis, because we're doing features. Number 4 is these homologous chromosomes are going to separate in meiosis 1, in anaphase 1. There are 2 cell divisions that we talked about. With no replication in between. This reduces the number of chromosomes. So we go from diploid to haploid. Now, when we talk about diploid and haploid, remember that diploid means that you have 2 sets of chromosomes, one from mom, one from dad. Haploid means you have half. So I can draw out steps 4 and 5 in our features of meiosis. So, when we talk about meiosis 1, we're going to say replication has already occurred. Your mom and dad's chromosomes have already been replicated. So the first thing in meiosis 1 that we say is you have synapsis. In meiosis 1, in meiosis 1, you're going to have synapsis and crossing over. Also, you're going to separate homologs. So in meiosis 1, what happens is you separate your mom and dad's homologous chromosomes. You're going to separate these, but you also have crossing over, which is going to look like this. So when we look at this, the first cell that we talk about is diploid. Diploid is one set from mom and one set from dad. Diploid. 2N, 2 sets of chromosomes, mom and dad. Mom plus dad equals 2 different sets, so this is 2N. So in meiosis 1, you now say that this side is your dad's cell, and you have your mom's cell, homologous chromosomes, so just a tiny bit of your mom's over there, and just a tiny bit of your dad's. So when you separate it, mom and dad's, this little tiny bit of the chiasmata does not count. So you start out with the diploid cell because it has one set from mom and one from dad, but you separate your homologs in anaphase 1. Separate in anaphase 1. So now this is 1N for your dad, and this is 1N for your mom. So in meiosis 1, you create haploid cells. Haploid means you have one set. So this cell over here, just say this one has dad's. So that has your dad's homologous chromosome that got separated. And this one over here is also haploid because it's your mother's information. So in meiosis 1, you created haploid cells. Now, these are called sister chromatids. So in meiosis 2, you're going to separate your sisters. So if I covered this up, those are sister chromatids of your dad. So now in meiosis 2, in anaphase 2, you're going to separate sister chromatids. So this cell has your dad's genetic material only. This one will have your dad's genetic material with a tiny bit of little baby mama information. This one has your mother's genetic material only, but you're separating the sister chromatids. And a little bit of mom's with a touch of dad's. So when we talk about meiosis 2, in meiosis 2, you're going to separate sister chromatids. Now, when we look at these, all of these are considered to be haploid cells. This one has your mother's genetic information. Your mother's with a little bit of dad's. It's still only half the information. It's not a full set. So haploid means it's got a full set of moms. Well, this one's a full set of mom. This has a full set, but it's mostly mom. So meiosis 2 creates 4 genetically different cells. And that's important because in meiosis, we're creating 4 genetically different sex cells. If these were all identical, we would look just like our brothers and our sisters. So meiosis, features of meiosis, it's important to understand how it goes and the definitions. So make sure you look over this. If you need to, Watch it several times.